Good morning or good afternoon, function where you are located. I'm really happy to welcome you for the 7BRG webinar on microencapsulation by emulsification. We'll have two outstanding speakers, very dynamic person, and I hope you will really enjoy. I will ask you to ask the question only through the Q&A window, and use the chat only if you have technical problem, and then somebody will try to help you with the technical problem. You will be able, if you have problem with your connection, to see the podcast of this presentation, which will be probably available, let's say tomorrow or Friday from YouTube, and I will send you a link to connect it. You know probably already, if you follow some of the webinar, that this is organized by two entities, the Bioencapsulation Research Group Association, which is one of the most dynamic group in microencapsulation, and you can get many information about it on bioencapsulation.net, and by an European project which is connecting a lot of people around the world. And you have also here the link to connect and get more information about this website. Before to start, I would like to explain you why we feel that this webinar is really important. And this is two work I done in the 80s, then 30 years ago. Then we are looking for the narrow side distribution for quite a while. If you look the control release of a drug from many papers, you will find a graph which is similar to the one you see here. And they are generally expecting that you have a burst effect, which will be a little bit of drug, which is on the surface of the particle. But when I read more carefully the paper, I observe that the burst effect could be 40% in the simulation I did here, but it can go up to 90%, and something is wrong. And in fact, I demonstrate, but not publish, that this is more a question of size distribution than a real burst effect. I also work on the breakage of capsule, and I arrived to the conclusion that the breakage is proportional to the diameter exposure 4. And then if you have a size distribution, you will have, in fact, a lot of problem that some capsule will break at some stage. How to get a narrow size distribution? One approach is the dripping technology that many of you know, probably, and it will be probably a webinar later on this subject, but today we'll have in fact two approaches. One which is in fact the membrane emulsification, which will be presented by Marijana, and a multi-canal microfluidic, which will be presented by Alessando. Then let's start with the presentation of Marijana. Marijana is working at the University of Loughborough. She got a PhD in chemical engineering. She has a strong experience in development challenge for the food, pharmacy, catalyst. She's involved for quite a while in the microencapsulation and try to make microsphere, capsule, and so on with very different format. And she is probably the starting of the company Micropore, which is developing quite a good equipment for producing, in fact, narrow size capsule. I will, in fact, let you follow his presentation.
I'm really grateful for the opportunity to present a little bit about membrane emulsification. So I'm Marijana, Dr. Marijana Dragosavac, and I'm a senior lecturer at Loughborough University, but also Chief Scientific Officer at Micropore Technology. So I want to kind of give this as a slide just to make everyone smile, and I think we all needed a smile at this moment in time. I'm originally from Serbia. I did my undergraduate degree and also my bachelor degree in Serbia under the supervision of my professor there, Milan Sovilj. I'm a chemical engineer from the start, and I really like to point that one out really often because chemical engineering does quite a lot for like scaling up things and making things into real process. As a part of my master, I came to do a short project at Loughborough University, and that's where I met my mentor and my dear friend, Professor Richard Holditch, with whom I actually did the PhD. I also did a postdoc as a part of my what we call knowledge transfer from university to Micropore. And then I was offered an amazing opportunity to start to be a lecturer and actually inspire the younger generations to do actually what I do and manufacture particles. So you can actually see me here kind of like riding a horse, but very often I feel as kind of like this purpley postdoc still with some successful research grants and also quite a bit of industrial experience. So I'll tell something about the Micropore. I think we started in 2003 and it was Richard Holditch who founded the company. And since then, it's been going from strength to strength with Dai as our CEO at the moment. We also won a number of awards in pharma applications. So if you want to know a little bit more about Micropore, I would encourage you to go and look online on that. But I'm going to focus my talk today more on manufacturing the particles, but maybe a little bit from the university perspective. But if you want, and we would be interesting to know more about the scale up, I will tackle that a little bit, but happy to kind of like answer maybe after any questions you might have. So when we think about the particles, very often they're quite complex systems, and I'm going to focus my talk today on a really kind of like large particles. We can manufacture nanoparticles, but I just kind of like, due to the lack of time, I just thought we might actually focus on the micron size at this moment in time. So when you think about the complex manufacturing, very often you might want to have something inside of your particles that is really valuable and you really want to protect. And something like that might also need an additional method of manufacturing. And both Alessandra and myself today will talk a little bit about that. So that's something that I would really want to stress. If you want to learn more about the nanoparticles, head on to both my website or Micropore and you can learn more there, but it's only about the micron size at this moment. So when you think about the conventional methods, when you think about the ways to make the particles, if we're focusing on emulsion-based routes, I've just shown here a few of those, and basically these devices apply more energy, and what will give you in the end is quite wide size distribution. They would be very good if you want to make tiny particles or drops, but if you want to make larger drops, and if you want to have something inside of those big drops, these devices are going to be really a problem for you, because very often what happens is that the big drops really get broken down into the smaller ones. So that's something that I think it's really important to kind of like be aware when you're trying to design larger drops. So having said that, what we really have to think about are different and maybe novel methods, how you can actually make those drops by drops. And I'm just showing here on the left side, something that I did as a part of my PhD. I had an experience to kind of use quite a number of different devices. So this is a microchannel, does create really beautiful drops, but the issue is that you can't really control with that one microchannel or microchip, you can't really control the drop size with the same device. What you could do, you could maybe get some with maybe smaller particle size, but still it would be really challenging. What I'm also showing here on the right side, and I'm hoping that you can see the video, I don't know, but I can't see if I can see some thumbs up so that you can kind of like see that there are some drops formed on the membrane surface. And what really happens is that drops are being pushed out and then squeezed out by the neighboring drops. So this is when there is no shear on the membrane surface. With quite a lot of, well, not quite, but actually really small amount of the surfactant in the continuous phase. So that's a really good thing when you think about drop by drop manufacturing of the drops, is that you really need so little amount of the surfactant compared to maybe the conventional ways, because of that surface and then good diffusivity of the surfactant toward the surface itself. The devices are really useful because they can provide you, they use very low energy. And when we think about the sustainability perspective, that's really something that it's amazing opportunity, but also they're going to give you very uniform size distribution. So when we think about the membranes that we use at Loughborough University, but also with Micropore, they are metal membranes. So you can see on the right side, we have nickel membranes. We have different shape of the pores because there are some advantages. Membrane, as every kind of like membrane, will eventually behave as a filter medium. And that's something that you don't want. So sometimes if you have nanoparticles or small particles as a part of your dispersed phase, you might need to have different shape of the pores, which would actually prevent and help you in a way not to kind of block the pores. But we started off with the nickel membranes, but due to the more pharma and food applications orientations of the micropore, we swapped to stainless steel membranes that you can see on the left side. And the very good thing with these membranes is that they have straight through laser drilled pores. So that's really useful because they can provide with a high flux, and that's really something that it's really super useful. Anyone who worked with the membranes very often might kind of like assume that they are kind of like membranes used for the filtration or cross flow with like interconnected pores, but these ones would not be something like that. They would have straight through pores and would make quite high flux. So as I said, in 2003, Micropore started with something what we call a dispersion cell, and that's something that you can use in the lab on a small scale. And we can actually generate quite, depending on the pore size, and also we actually have the rotation of the pedal above the membrane. And I hope you can see my mouse pointing to the membrane here on the left. But as we increase the rotation speed, our drops are actually decreasing. So we started with that, but then now Micropore is manufacturing a cross-flow units that actually have quite high capacity. So they can go up to 1.5 tons per hour, depending on the formulation. And again, as we increase the flow of the continuous phase, we can increase the shear, we can change the drop. So with the same membrane, we can actually now create different drop sizes depending on the shear that you want to kind of like operate at. So if you want to get in touch with Micropore, feel free. Then I guess Dave or someone from there might kind of like help to kind of see everything from the perspective of the scale up, but also in Loughborough University, we have all these devices, so that makes my life easier if I want to do the formulation work. So that's quite useful. So I'll just quickly tell you, I don't like to kind of put so much modeling in my presentations. There are conferences where I would do that more in depth, but I thought to just give you a flavor of what actually membrane emulsification and these particular devices with the metal membranes can actually do, maybe if you're thinking about your formulation. Because as a chemical engineer, you really want to know what the device could actually provide you. And we do this particular experiment also with our undergraduate students. So you have a drop, the oil is pushed through the membrane, and it's the capillary force that actually tries to keep the drop on the membrane itself. So if you have your formulation where you can go and you can measure your interfacial tension, you can look what actually pore radius is available, and that will give you an idea about that capillary force that will keep the drop on the surface. What you can also do, you can apply certain shear on the membrane surface that will cut off that drop, and that drag force is a function of the shear that you generate on the membrane surface itself. As the drop grows, basically at the moment when that becomes equal to the capillary force, that's where we assume that the drop is detached from the surface itself. So that gives you an idea, if you know your formulation that you're maybe working on, if you know the interfacial tension, you can play with a shear and understand what drop sizes maybe you're able to achieve with the formulation and membrane emulsification. So I'm just kind of like here showing this, what I was saying. If I look different methods that we kind of developed as a part of the micropore and university work, you know, we start from the dispersion cell and we look for the model, and we can see kind of like nice same behavior of what we can actually generate using different productivity methods. So that's really useful to be aware that really a simple model can explain. We could do CFD, but I don't think it's needed at this moment in time. So this brings me to the time, what I can focus my talk a little bit more on the formulations itself. Number of the formulations have been done within the labs, and I think that's really amazing. It's not one person job, but a number of the students have been doing it over the years. I'm going to focus my talk today on the cell encapsulation and also the biodegradable particles, because that's something that we have been quite proud of. But also, if you want to learn about inorganic materials that we can manufacture or a number of different formulations, feel free to look Micropore website, but also number of publications that have been done over the years from the group that started Professor Holditch, who retired a few years ago now. So all kind of like does the same. I think I really like to, at this moment in time, appreciate all of the students that helped really build the research that I'm actually working on and continuing to work. And I will just, say big thanks to Serena. She did an amazing work. She came from Italy to do her PhD on the cell encapsulation. And it was really interesting experience to start, because I think she was more someone who was related to pharmaceutical applications. And then we had to work out, as a Chem-Eng department, we wanted to kind of like do a scale up and things relevant to that. But the idea behind the work was to try and get the development of the pH sensitive particles. And having said that, we were thinking to try and look how we can actually use membrane emulsification, do the formulation work, but then also do the scale up of those kind of like cells being within the capsule. So that was kind of like aim. But the intention also beside that was that we were trying to kind of like generate the particles. They would kind of survive the stomach, so they survive the acidic region here, but then really kind of decay in the stomach. So all about trying to get the probiotics really in the place where they kind of needed to do. And it is quite a bit of formulation that has to be thought through, because if you look at the literature, very often number of those formulations are done maybe, if you think about the dripping, or it might be different ways where you can actually have quite high viscosities of your polymers. But then when you're thinking to put it within a new process, you have to, think maybe about the viscosity, density, and so many other things. But we looked some kind of like cheap material. And I say cheap material because when you're a new lecturer, and I think Serena was my first PhD student at the time, you really want to save the costs. You don't really have huge amounts of money to splash on stem cells and so many other things. But she actually sparked quite a lot of extra research. And in the lab, we actually have Lisa at the moment, who's actually trying to encapsulate the stem cells. But we started really from some cheap material. So let's give you a flavor of what we did with Serena. This is just a part of the formulation work. And what we had here, we had dispersed phase, and Serena did a number of formulations. So she did alginate, chitosan, gelatin, but that dispersed phase actually had the cells inside. So this small device, dispersion cell, can rotate, and basically that rotation cuts off the drops from the membrane surface. We can then look how, as we increase the shear, so on the right side, you can see as the drop size, how the drop size is a function of the shear. And we can see as we increase the shear, the drops decrease further down. And that's something that I've explained with a simple model. And you can see that if I look also the coefficient of variation as a function of the shear, we are quite below that 40%, but also, you know, we can go down even lower. So I think for us, it was really useful to try a number of the formulations, because we learned quite a lot, you know, to kind of do different, yeah, as I said, viscosities and what actually is doable with this particular type of equipment, and test it to its limits. But we can see that, yeah, we can control with the same membrane. And that's really something that is really useful. You know, you don't change the membrane itself, but you just change the conditions of the operation. And having said that, you can generate different drop sizes with quite uniform particles. As I said at the start, I think the biggest issue with if you want to push something through your membrane, you every time are doing a trade-off, because there will be a certain concentration that you can work toward as cell-wise here, that you know, your membrane will start to behave maybe as a filter medium rather than a device that will actually create drops. And to be fair, everything that has a nozzle or something would behave on the very similar way. But there is kind of the trade-off to understand maybe, you know, what are the polymer concentrations and also the cell concentration that you can work toward to actually get that good size distribution still. So we knew that the probiotics, if we want to kind of like think of this as a probiotic application, we need to work on cell concentrations up to 10 on the power of 8. And what you can see on the slide here is production of that water in oil emulsion, where the water phase, these particular photographs are from gelatin. They do have tiny dots inside. They are actually the cells, and you can see that the concentration of the cells increase. But the good thing is that even the concentration of the cells increase, the drop size stays more or less the same. And this is with a 30 micrometer membrane. We can have quite good CV, and that's really, really good. So we succeed to kind of like encapsulate quite high concentration of the cells, and the membrane still worked without any issues. So then I would say that I was in a way maybe a terrible supervisor. So then I was really happy at that moment in time. But I said, okay, so that was very nice, but can you make them pH sensitive? So can you make them, you know, you can make those nice spheres, and you can make them kind of, you know, have those cells inside. But can you then try to kind of get an additional polymer around? So this is where kind of, I would say, Chem-Eng degree that she got, and I'm trying to kind of like teach also my students, is that, you know, there is an additional things after you create the drops, how to actually make them into solid particles, and then how they can actually do the trick of whatever you want them to do. So she succeeded to kind of like develop a method where she used U-Dragit, pH responsive polymer, to coat the particles, and we were able to succeed that the cells would kind of like survive the acidic conditions, but they would release on the higher pH. And we could observe that using confocal microscopy. So the green cells would mean that they're viable, and red cells that they are dying. So if we release the cells, we can, you know, understand that we can count them. But also these would be kind of like a bit more fancy, I would say, way of confocal microscopy. The other reason why we went for yeast cells to understand the success of our process was also there was a very good and easy way to detect the viability, because the cells, in order to grow, they can consume the glucose. So that was another way for her to track also that the glucose was consumed with the time. And we showed that that was the same for number of the formulations, but also number of the concentrations. Because when you think encapsulating cells inside, and I'm sure, you know, there are a number of you working with that. field, you understand that also the cells manufacture also something that might kill them if it's kind of all encapsulated in everything, but also you have to manufacture the 3D environment that would actually be permeable for the gases that they might produce, or kind of like waste that they might produce, but also enough for the nutrients to diffuse in. So we were really happy to kind of do something like this for a number of the formulations. So I think I'm not really sure how I'm standing with the time, but I will just tackle another formulation. This has been an amazing visiting student, I would say, Alessandra, who is currently in Germany. She also came from the same group where Serena came from, University, CNR in Cosenza. And I think her work was really useful, because what we were trying to do with the work that she undertaken is that we were trying to understand how we could encapsulate both water-soluble and oil-soluble compound within the same particle, and also use membrane emulsification, because we were trying to achieve very high encapsulation efficiencies with very low amounts of the polymer. And the one thing that I really want to mention here is that very often when you think about maybe injectable formulations, they're normally made maybe in a very in a high shear mixers, and they don't really care about what is the viscosity, what is the density of the material, because they can chuck everything inside and then they disperse everything around. But here, when you think to push something through the membrane or to push something through any channel, you have to be really careful about what are the maximum concentrations of the polymer that you can work. But that's actually a very good thing with also, and I will show you that in a second, because then you can play with how fast you solidify that polymer. So you can actually save money by using formulations and these drop by drop manufacturing methods. So quite a bit busy slide, I would say here. So I'm just going to guide you a little bit through. On the left side, what you see is just different setups that Alessandra used. So we had a dispersion cell where we had an oily phase with dichloromethane polycaprolactone injected through the membrane. And we were creating those particles here. We tested a number of the formulations with some, I would say, additional materials inside of that polycaprolactone. And also we tested different surfactants outside of those particles in the continuous phase. And then she also did the scale up. So she used azimuthal oscillating system, but after that we also used the cross-flow systems to do the same. So what you can see on the slide, on the images on your right side is again very same, as we did before. You will look for the drop size as a function of the shear, and what we can see is that with the same membrane, what you can produce is that you can make particles between 100 and something down 50-55 microns with the same membrane, and quite a low. So unfortunately here I'm using span as a way of uniformity, so that would be D90 minus D10 over D50, and it's those particles are really down to 0.3 as uniformity, as you can see. We can also look how the injection rate, so you have the same shear, but you change now injection rate, and then we can see that injection rate, as it increases, we can actually have that drop size also increase. So it just gives you the flavor. Again, the same membrane, capable to produce a number of different sizes, which is something that might be really, really useful. So I would say the pore size was 20 microns. I don't think I've actually written down anywhere, but micropore manufacturers now the membranes down to 5 microns. So you can actually see that we could actually make particles smaller than what actually has been shown here. What also we try to kind of like do is I say it's very nice when you do a publication, and then in publication you kind of like show the best possible results. But I remember Alessandra at the time, you know, she did really hard work and tested a number of the formulations. But the intention was to try and use a small water-soluble molecule. So we went for copper because it diffuses really easily into your water phase, and that's something that you really want to kind of be careful of. The intention was to try and understand how your surfactant in the continuous phase can also assist in your solidification of the particle itself. So when you really think about that, what she tried was sodium dodecyl sulfate, and she also tried PVA, polyvinyl alcohol, 1% of that. And she also tried different ratios. When you make the drops, how the ratio will affect your solidification, if you want to call it like that. And we worked out that you need to work if you want to have that water-soluble compound inside of your polycaprolactone particle, you have to at least have 1:5 ratio of whatever your continuous phase to what you mix it with to create that flash evaporation. And also then, you know, you can actually see that the encapsulation efficiency of copper was quite high. She also did the tocopherol, what was really useful with tocopherol to understand is it's a lipophilic compound, and actually it stays inside, so it doesn't surprise us that that's really high. So here the challenge was to actually have very high encapsulation efficiency of copper particles, and actually, you know, we proved that we can keep that really high up. And the very, very important thing to note is that the concentration of the polymer of that emulsion was really small. So we were working with 10%, maybe 20% of polycaprolactone, where normally if you would do that in a homogenizers or stirred vessels, you would have to use maybe 60-70%. So we succeeded to kind of like reduce the amount of the polymer really a lot. And that actually really helped us to kind of move on to a more kind of injectable type of the polymer. So we did, after Alessandra's work with, we also did some extra work on PLGA. And we looked also to reduce the amount of the polymer that we should use. And we succeeded to kind of create, we looked into very costly and pricey peptides for specific cancer treatments. And we succeeded to have very high encapsulation efficiency, where normally you would want to have this above 80%. And that's very common if you use very high concentrations of the polymers. And we can also see that we can see here that our encapsulation efficiency is still high, 85%, but very low amounts of the polymer of the PLGA used in this particular case. We can also control. I can just show you that you can see that there is a surface of these particles is also different. So there are small tricks of trade, I would say, if you want to keep them this smooth, but having this water in oil and water emulsions, there could be some additions in the continuous phase that you can make this as a smooth surface if that is something that you needed for the formulation as an injectable. So if you want to kind of learn a little bit more about that, feel free to get in touch, and happy to have a chat afterwards. So very high encapsulation efficiency here, and with the reduction of the polymer. So I can't really track, unfortunately, the time, but I hope that I'm in half an hour. I would say that it brings me to the end of the talk when I think about the bioencapsulation research group and particles that we done. These are just tiny bits out of like number of the particles that we did and we plan to do. But I hope that you got a flavor what we could do at Loughborough also now commercially through Micropore. we can get that very high productivity, and we can say that the membrane emulsification can be, reliably now scaled up to really high values. There are quite a bit of particle sizes that we can do. So I will just stress again, what I've talked today, they are micron sized. I have not talked about nano. That's another topic, and happy to have a chat about them at some point. But here we can easily make particles from 10 micrometers and above, and some formulations we can go even below if needed. But I would like really to leave a little bit of image impression that I like to give every single time at the end. If you want to encapsulate something and you use homogenizer, and that something might be, for example, water soluble, you can see that what you make here, even just by optical microscopy, you can see that the shades of gray, 50 shades of gray, are completely different. So that means that quite a lot of your material, and very often that's very high value material, went out of your drops. But if you're using membrane emulsification, and if I zoom this, you can actually see that you pack really nicely material inside. And I can say that every formulation can be adjusted to membrane emulsification, but everyone who is doing that, they just need to have time. But also it's not only making the drops, it's quite a lot of process that comes afterwards. It is that difficulty to make the drops into particles that makes my kind of like research really exciting. So I'm not going to finish by, maybe not inviting, potential students. You might want to come and stay as a visiting researcher, as Alessandra did, for example. But, we would be very happy to, if you have a formulation that you want to try, and you're a student or a postdoc, feel free to come to university. If you're a company, feel free to go to Micropore and talk with them about the scale up. But I'm very, very interested and have quite an active research, not only in the particles for the drug delivery. At the moment in the lab, we have few that are working on ion exchange resins and also novel catalysts, because this method actually provides you that opportunity to really put wherever you want things inside of your particle. And at the end, nothing of this would actually be available to me if I didn't have my supervisor, Milan Sovil, initially. I then met my another supervisor, Goran Vladislavljevic, who also helped me to come to Loughborough. But then I met Richard Holditch, and I really enjoyed working in membrane emulsification since. A number of the researchers went through the lab, and also a number of the funding, and I'm really happy to continue the research. So if you want to support the research, feel free to get in touch, and I'm happy to have a chat. So that's me.
The second speaker is Alessandra Hofner, which is still young in the domain, but already make a great job. And he will present the technology which is he developed during his PhD. He has created a company, and this company is growing quite fast. And you see that he gets a different price for his technology. And I hope you will enjoy, in fact, his presentation. And I hope this time I will not freeze again. Then I hope you will enjoy this presentation.
Welcome, ladies and gentlemen. My name is Alessandro. I'm the co-founder and CEO of Microcaps. And in the next couple of minutes, I would like to show to you the evolution of microfluidics and how by upscaling microfluidics, we're able to engineer and innovate new ways of microencapsulating products. At Microcaps, we're working in a wide range of industrial applications, from cosmetics and personal care over novel foods to pharmaceutical applications. And we bring with our approach different benefits to each of these industries. So for the cosmetic sector, it's mostly about visual enhancement and sustainable products, so reducing microplastics waste. Novel food, it's mostly about encapsulating flavors and fragrances, but also going into novel and living materials, such as cell encapsulation or probiotics encapsulation. Versus in pharmaceutics, it's all about new release profiles of actives and control and tailor the release over time. But actually, what's the goal of this presentation? The next couple of minutes, I would like to prove to you that microfluidics is scalable and ready for industrial applications. And second, I would like to present to you novel formulation opportunities and really novel opportunities for new active encapsulation. For that, I have a picture on the right-hand side, which should combine number 1 and 2. So these are actually oleocaps, an oil core surrounded by an alginate gel, which we're producing at high throughput. This is not just a droplet by droplet experiment that was possible under the microscope, but it is actually an upscale technology that we're producing at the moment. And number 3, my third goal of today, I would like to invite you in the end to think about new opportunities for end customer products and actually really take the microfluidics as an example. So now having this in mind, what we can do and actually think of new products that are now possible. So first of all, what are the obvious benefits of microfluidics? So if you compare it to today's methods, state-of-the-art methods, such as shear emulsification, batch emulsification, or membrane emulsification, you end up with very uncontrolled capsule size, therefore very uncontrolled release profile. Versus at microfluidics method, what we're doing at Microcaps, you have all capsules with a very uniform, such monodispersed size distribution, and therefore all are acting exactly the same. So you can tailor and shape your release profile accordingly. Additionally, you have a very consistent loading of your microbeads, so all are exactly the same. So meaning that you can control the dosing in each individual capsule, not only in the individual capsules, but also batch to batch, you have always the same reproducibility, and you can have a very consistent performance throughout your production. You have very low active waste, 90% plus of your active in the end is in the capsule itself and not just somewhere in the continuous phase. And also in the end, you don't need to filter or size select the right size, but you can actually directly target exactly the desired size that you want to have. And you don't have any stresses involved, so no shear forces, which allows you to work with living material, with complex APIs, etc. Now, the problem of microfluidics in the last 20 to 25 years was really the lack of scalability. And how we overcome this, I would like to show to you in the evolution of microfluidics. So we start on the left-hand side with the shear-induced devices. We have either the glass capillaries or T-junctions or flow focusing, where we actually have a dispersed phase, which is then sheared off by a continuous phase. All these devices work in the same method. You have a continuous phase which shears off a dispersed phase. Now, the size of your capsule here is dependent of the shear force of the flow rates that you're actually bringing into your device. And while at the beginning we think that's a huge advantage, I have one device and it allows me to create different sizes of capsules. In the end, it isn't. If you think about upscaling this and putting now not only one of these droplet makers, but actually already 2 of them, they are linked together, meaning that if you have pressure perturbation, which you always have in microfluidics, and you increase the pressure on one channel, the other channel will be lower, and therefore have a different size of capsules. That comes the same if you have some dirt, if you have some clogging, you will actually increase the pressure on the neighboring channels. And so overall, if you collect them in a beaker over time, you end up with a very uncontrolled mess, and all the benefits are simply gone. So you just sit there in front of your microscope and you hope and pray that you end up with gram samples, which you can then actually look then under the microscope. Contrary to that, we have step emulsification, the method that I showed to you here on the right. In step emulsification, you have a tiny channel of your dispersed phase going over a nozzle to a sudden increase in height, the so-called step. And this step is actually gives you a physical reason why the droplet is actually detached from the dispersed phase channel. It's a Laplace pressure difference, which sucks the droplet into the step, into the continuous phase. The beauty of this is that the size of the capsule is independent of the actual flow rates that you're bringing. So one device gives you one distinguished capsule size. And this is a beauty because now you can start parallelizing those droplet makers. We started at the beginning with 10, went to 100, went to 1,000, and now are reaching 2 million of such channels next to each other. And even if 100 of them gets clogged, it will increase the pressure on the neighboring channels, but the pressure increase won't increase the capsule size, meaning that you can just walk away. You have a very robust system, very robust microfluidic system to produce actually your capsules. And to prove this, I have here an image where we do something very complicated. We're encapsulating double emulsions. So we have an emulsion, a droplet in a droplet that we're producing in these capsules. And you already see at the end of the nozzle, you already see some fouling that is occurring. That doesn't affect at all the capsule size. And that's the beauty of it and why it's so robust and why it's here to actually upscale this method. So if we look at what we're doing with our EOS system, emulsification optimized system, we have the ability to scale up 750 kilogram per day. We have everything in glass or stainless steel, so it's very chemical compatible, and the consistency of performance is very high. So if you have fouling, that doesn't affect the sample quality. If droplet makers get blocked, that won't change anything, meaning that you have very minimal maintenance. You can just exchange also your chip, meaning you have your sizes of capsules with one equipment. And beauty of microfluidics, you have very reduced solvent use. So typical droplet to continuous phase ratio is at 1:1 or 1:2. This is compared to 1:50 for batch processes. And again, to show you a video, this is how step emulsification works. So high throughput and very nicely. The EOS platform that I just explained now, we have two systems. We have the innovation system and the pilot plant system. The innovation system is a compact laboratory-based device, which is based on 5 centimeter chips up to 100,000 junctions that allows you to iterate. So for example, if you want to test different level of your actives, you use your innovation system versus the pilot plant that is a large scale device, which uses 20 centimeter chips up to 2 million junctions and can produce actually up to 750 kilograms per day. Droplet size range and capsule size range always between 20 micrometers and 3 millimeters. This is in the end how a size distribution diagram looks coming out of EOS. So we're very proud to always be between 3% and 5% versus best state of the art is at 25%. This is on size distribution. And now I will actually show to you how our EOS actually works. So this is at real time. We just open, we just press the button and everything works at real time. So these are 100 micrometer droplets. And then we just stop. So you see the massive throughput that we brought and the upscaling of a microfluidic method. At Microcaps, we're not only working on the novel equipment that I just showed to you, but we also want to introduce novel formulations that are now the benefit of microfluidics. We're working with solid caps, thermocaps, oleocaps, and hydrocaps. So solid caps are using solvents which are extracted, most of all PLGA systems. Thermocaps is fat or hydrogenated oil capsules, which we heat up and then thermally quench. Oleocaps is a liquid oil core surrounded by a polysaccharide shell. And hydrocaps is a polysaccharide matrix bead that looks as following. We have sizes between 60 micrometers up to 3.5 millimeter. You can use alginate, PGA, agarose. You can use this to carry cell growth medium, but you can also encapsulate probiotics and bacteria, or you can use it to carry fragrances. Versus the oleocap system, now we were back to the capsule that I showed you at the beginning. We have an oil core, liquid oil core, and we grow an alginate shell on top of that oil core. We have a size range between 300 micrometers and 3 millimeter. And it's beautiful because you can have a visual enhancement if you want. You can encapsulate different types of actives, fragrances, flavors, but you can also introduce probiotics in a powder form into the oil phase. So you never expose them to water, so they just stay in the dormant stage. And you have a very nice burst profile. You can just pop up the alginate gel and you have the release of your oil containing the active. So now on the third side, I would like to show to you different examples why customers are actually working with us. So in pharmaceutics, it's really about controlling the release profile of your active. You can also increase your release horizon by 100%, so just doubling it. It's a lot about taste masking. You can enable new and novel therapies, for example, LBPs, so using anaerobic bacteria for actually pharmaceutical treatments. And you have the benefits is you have very precise dosing, very controlled release profile, and very batch to batch, always the same type of capsule. In cosmetics, customers work with us, first of all, to replace microplastics, such as, for example, PVA. So all our capsule systems are sustainable, biodegradable. You saw the capsules before, they're beautiful. So you have visual enhancements. You can put shiny glimmer into your capsules. You can have a pearlescent appearance through titanium dioxide. You can have texture enhancement. And also here, encapsulate fragrances. You can have controlled release over time or a triggered release. Or you just name it. The field is open for your imagination. Why do customers work in novel food with us? It's about enhancing bioavailability. So you can encapsulate vitamins, you can encapsulate CBD. As well here, you replace microplastics. You can also have all sorts of vegan, halal capsules, so no gelatin anymore. And you can also increase your probiotics performance through encapsulation. And in the end, you can just simply suspend your capsule in, for example, water and have a very nice product where your capsules swim in a beautiful drink, or you just name it. The field is here also open for imagination. So that's it from my side. I think the goal of this presentation was to prove to you that microfluidics is scalable. I hope that I was able to convince you that we have new and novel formulations opportunities, and we can actually encapsulate different types of actives. And I still invite you to think about new products that could come onto the market. If you have something, please contact me directly, shoot me an email, give me a phone call. And let's discuss this. So this is the team that we're having at Microcaps. We're based in Zurich in Switzerland. We're currently 20 people. We're actively hiring, so we have 4 positions opening. Also, if you're looking for a new job, please come and talk to me directly. Or if you want to discuss your microencapsulation project, we're very open and very happy to start a discussion with you. Thank you very much.
Okay. I hope now we can continue without too much problem.
Welcome, ladies and gentlemen.
Okay, I will propose to Alessandro and Marijana to join us in a way that we can probably answer to a few questions. I think Alessandro will come soon. I think she already answered to Arnaud Picot, but maybe I will be interested to have also the answer. Marijana, how do you manage to clean the capsule after you make water in oil emulsion?
Well, I think it will really depend on how survivable the cells. I think there was a question about the yeast cells. So I think the more sensitive the cells are, I would start with maybe very kind of like gentle surfactant solutions and then wash it with that. But if the cells are more more rigid, as maybe the yeast cells would be, you could actually use some of the solvents and the cells would still be viable after that. So it's just really down to the cells that you have, what's the process that you use afterwards.
Okay, then Sophie, congratulate you, but I think everyone congratulate you for your presentation. And she's asking, can membrane emulsification prevent agglomeration, flocculation of the droplet when inducing shell formation?
So I wouldn't say that the method as it on its own, it can actually prevent something like that. It just helps you to maybe generate drop by drop, but it's actually, you know, fine formulation afterwards that actually can provide that. And also, if there is a need to have that not contact close by, there can be design of the membranes can be changed. So you can actually change the spacing between the pores, and in some formulation that helps to prevent fully. But I would say if your drops are sticky by its nature, there are methods to kind of overcome that by adding maybe an additional surfactant, or what we would normally do, you could maybe even mix with some particles that would actually coat to the surface, something like a Pickering emulsion to that, but then it's a little bit more of formulation afterwards. But yeah, if you need, I'm sure we can get in contact and have a chat on that.
Okay, Jenny is asking, what is the smallest particle size we can be achieved? How important is the effect of viscosity on continuous and dispersed phase?
Maybe Alessandro can reply to that and I can follow.
I think it's maybe for both of you. I think Jenny, it's probably interesting to get the answer from both of you.
So yeah, I think you can go, it just really depends on the formulation. So if you have a flash evaporation, if you're trying to make like nanoparticles, you can make them, but it's a different dispersed phase and continuous phase. So you can make smaller particle sizes, but with membranes, with those kind of like that I was saying, now, maybe like 5 to 10 microns would be a push with the formulations that I was talking now. So I think if some other formulations would be in question, then you can go without issues, possibly even below the micron size.
Yeah, and from our end, it's also similar. The 5 micrometer is a very good limit, especially for the solid caps that I presented. So PLGA capsules would be at 5 micrometer versus like alginate type capsules rather at like 15 to 20 micrometers. It's actually not a technical limit that you're having there. It's really a question of throughput. So if you go lower, you can actually produce kind of channel sizes that are smaller, but then you're at a certain limit where actually the throughput massively drops down, which is why we're kind of stopping at these lower ends. So yeah, 5 to 20 micrometer, that's kind of the lower end, I think, of both technologies, mainly because of throughput reasons.
Okay, Talita asked a question which may make a fighting between both of you. She asked, in fact, what is the advantage and disadvantage between the membrane emulsification and the microfluidics?
I don't think that there is kind of a competition. I think, and I replied to her honestly, or to him, I think it really depends on the scale that you need to produce. And it's going to really depend on how much you can sell your particles for and how much of a trade-off you can give for the CV. And I think, if you can go down to, if you need to go, below 5, then there might be some trade-off between different formulations. But if you look what we can do also from the cross-flow perspective at the moment, that can go down to 5% also in a CV. So it's really going to depend on the formulation, I guess, and the cost on.
Yeah, I also highly agree on that. It's not a it's not a fight between the two technologies. I think it really depends on your end application. For certain applications, surely one process is better than the other, but I don't think it's a fight between the two technologies. size range is the one, then throughput is the other, then in the end it comes on the formulation itself. And there, yeah, it really depends on the end application.
Yeah. I have a question from Anand, which say, can you prepare B continuous emulsion with membrane emulsification?
I wasn't really I couldn't understand. What does it mean as a big continuous?
I don't know, but maybe Anand may ask you. I don't know.
Yeah, I mean, we do have like even the dispersion cell can be made into semi-continuous process. So we have like specifically designed, you know, even for the formulation perspective, you can test as a continuous. So we can have inlet and outlets. So you could modify it into a continuous process. But we have like, we had some work done as a part of my recent grant where we kind of like have a continuous process. We have oscillating membrane or cross flow, and then we attach with the reactor afterwards. So there is no reason not to make it as a continuous or semi-continuous, if that is what I was understanding. But I don't know, by continuous, like two, maybe you can do two in parallel. That's another thing that you could connect like few membranes in parallel, but I'm not really sure exactly what it means.
Okay, I have a question which could probably for both of you. Do you have experience with flavor essential oil and encapsulation? Then maybe Alessandro first.
Yes, from our end, quite a bit. We work a lot with fragrances and flavors encapsulation. So the oleo caps that I showed you in the end would exactly be the perfect formulation for that, where you have a liquid oil core surrounded by an alginate gel, and you can mix the oil core between, you know, normal MCT oil, for example, and your essential oil. And you can go to quite high loading, and we do this quite often. So this is one of the things we're very good in.
Yeah, and I would say like from Micropro's perspective, they have quite a bit of industrial experience with that on the scale up of the method of flavors encapsulation. But also in university, I mean, I was very fortunate to get quite a bit of different flavors recently. So my undergraduate projects, my undergraduate students would actually test some of the nice kind of like orange peel of the things. And, you know, that's kind of like how a big maybe project starts. So there could be some formulation done also, you know, on a university level on some novel kind of like works. So, you know, there is quite a bit of experience both on the university side, but quite a bit of on Micropore as an industrial kind of scale up.
Okay, we have a question from Kay. He's asking, you mentioned also use of nanocellulose as encapsulation material. Can you say a little bit more? What kind of nanocellulose did you use? And probably I will add a question on it. You feel it's something like the Pickering when you have really...
Yeah, you can kind of like thinking of trying to kind of like have like as a small fibers, and that it really depends. Do you want to use the fibers as a Pickering emulsion to stabilize it, or to have it as within side of the material? So there are different applications that were tested and could be done, but it's commercially available. Yeah, so.
Katarina is asking about high viscosity, but I think you already answered a little bit to this. One question that can be for both of you too is membrane emulsification, or the microfluidic favorable technology for encapsulation of anaerobic cells.
Yeah, maybe I start for that. Yes, absolutely. Anyways, for cells itself, I think all the gentle and shear-free emulsification methods in microfluidics are very well suited, because you end up with a very high viability of your bacteria in the end in the capsule. So we are at around 89% of viable bacteria in the end. And the other thing which is a beauty is that you don't have any exposure to air at any point in time. So you can actually also in the pumping system itself, we use pressure pumps. You can generate pressure there with you don't need to take air. You take nitrogen, for example. So that works very well. So in the end, you never have any exposure to air, and you end up with your your bacteria encapsulated in the capsule with never having any exposure to anything else, and they're super happy and viable in the end. So I think it's a very good method to work with bacteria in general.
You want to add something, Marijana?
No, I think
Okay.
I would completely agree. It just really depends how you formulate it, and if you do it within the nitrogen atmosphere, that would be kind of way forward to kind of not get it into any air connection, but it's doable.
Obviously, it will be an advantage in regard to a technology like the dripping or something like this, you know.
No, but yeah, I think it's down just really like on the product, you know, I think it's like every time thinking about what's the amount that you want to produce, how much it's going to cost, and really, you know, how bothered you are with your coefficients of variation of, I mean, I really love to see Alessandra's like images, like they're all the same size. And, you know, I think that's what made me kind of do a membrane emulsification too. So I think if you can make that in high quantities, you know, that's really good.
In fact, in some way, it's not only anaerobic, but it's to keep sterilized the system is probably more easy than other kind of technology. One question that can also for both of you is that was there any bead coalescent problem? Is there any solution to address it?
Yes, absolutely. I mean, coalescence is always a challenge when you have emulsion-based technologies, and you know, we work always with emulsions and then create capsules in a process right after creating the emulsion. So I mean, the trick behind this is really to stabilize well your emulsions. So using surfactants, using pickering, I think is the key. And if you manage to do that, you have them very stable, and then you don't have any coalescence until you create then actually a solid capsule out of it. And at that moment in time, you're perfectly fine to go. What you need to be a bit careful is that they don't start sticking together. So in the end, it's all about pre and post process handling. So the EOS system, what I showed you, is really the emulsification module, but the pre and post process, there's a lot of know-how and a lot of processing involved there. That you don't have coalescence right in the emulsification creation, and then no sticking to each other in the downstream process. And if you manage that well, then it's in the end a challenge which you can easily solve.
Okay, I have a question for Alessandro. With your technique, could you maintain the core in a liquid water status inside the microbeads? I don't completely understand the question, but maybe you figure out.
Yeah, I think it's about whether you can create, you know, capsules with a liquid core inside. I hope I get the answer, the question correct. So, I mean, the oleocaps that I showed to you is actually liquid core surrounded by a solid shell. And there's actually two ways of creating liquid parts within a capsule. One is what we call multi-core, where you basically have kind of a matrix type bead with a lot of little tiny core of liquid, and the other one would be the oleocaps that I just showed you, which have one single large big core in there, and depending on the type of capsule, you can actually change the release profiles. You need to imagine the oleocaps, they would just pop off, so it's a single burst and everything goes out, versus if you have kind of a matrix type bead with a lot of multi-cores inside, it's kind of popping up slowly over time. So it's kind of like you need to imagine kind of like an onion which decreases from which degrades from outside, and whenever you have a tiny little drop at the surface, it pops open. So over time you get a release over time. I hope that answered the question.
Okay, the last question, which is probably the most difficult one, is that in fact, what are the costs per kilogram, especially when compared to conventional techniques? The product is very nice, but is it over cost which will be justified by the advantage of the technology?
From my side, I think if you want to know any costing, I think you have to get in contact with Dave or Alex or with someone from Micropore, and they would be able to, reply on that. But I think it's just really down to, productivity and everything. But maybe Alessandro wants to kind of like reply from Microcaps perspective.
Yes, it's obviously a very good question. And now we were talking in both presentations in so many different fields, and depending on the active, depending on the field, you have different cost per kilogram. So if you look more, in a sector where a standard would be at around 25 euros per kilogram, this is obviously what drives us as well, and what we're reaching. But now again, it would be very unserious to say cost per kilogram now based on which application in the end, based on which capsule size and which active. What we are doing at the Microcaps, it's in the end what drives us. Obviously, our product needs to be able to compare to the market and to be able to justify the cost. It cannot just be better, but it needs to be also comparable on a price level. So we are good there, that we can compare ourselves with the market standards there.
Okay, we'll stop there with the question, and I would like to thank Marijana and Alessandro for the great job they did, especially Marijana for saving this webinar. If you have any more questions, you can contact them. The podcast will be available if you go back to the BRG website, probably over, let's say, 2 days. This is the end of the series of webinars for this series, and then we are thinking for the next series, and we need your help because we will organize the next webinar in function of your request. And you want us to talk about some technology, some process, about the scale-up from some application or domain, or do you feel that we have to talk about the characterization or analytical method? Then your suggestion is welcome, either through the BRG website, or you can send me a mail to info@encapsulation.net. We still have some events which is in the program. The most urgent one, if you want to attend, is the Microencapsulation Industrial Convention, which will be an online in June 10. It's mainly a business-oriented events. Then you will have one-to-one meeting if you want to discuss with people, to make collaboration, to find supplier or to find customer or something like this. It will be another one which will be on place in Rotterdam in May 22. And we are thinking to organize more scientific events in the spring 22. You will probably get invitation for these different events, but if you want to follow, you can go on the Bioencapsulation Research Group website. And if you yourself would like to organize something, you can contact us too. Thanks a lot for attending this webinar, and thanks a lot to Marijana and Alessandro, and we hope to see you again very soon. Bye, and see you soon.